the mainstay chemotherapy that is given to patients is temozolomide, and temozolomide is administered as an oral capsule. And indeed, almost every clinical trial which has tested new drugs has delivered those compounds systemically. So this is either through the bloodstream or taken through an oral capsule. There's a number of challenges that presents. The first is that as you're delivering the drugs systemically throughout the body, you are exposing healthy tissues and organs to that toxic compound. So that limits the therapeutic dose that you would like to ideally get into the brain due to toxicities to the tissues and organs. The second, often insurmountable challenge is that our brains have evolved a protective barrier called the blood-brain barrier. And normally this is a fantastic evolutionary adaptation because it stops pathogens and microorganisms getting into our brain. It's very rare to hear of infections in the brain because of this reason. But when it comes to delivering chemotherapeutics into the brain, this barrier either impedes drugs getting through, or it doesn't allow compounds to get through as effectively. And so the majority of drugs that we deliver either don't cross the blood-brain barrier or they do so very poorly. And so there you have this real challenge in trying to get not only drugs into the brain, but then for those drugs to penetrate through brain tissue to get to where the residual cancer cells remain. So based on this, a related aspect to the work we do at the University of Nottingham is once we identify what new compounds to use, we aim to deliver them during neurosurgery. So when the brain is open and towards the end of the operation, there is a window of opportunity to deliver drugs, what's called locally, in close proximity to the residual disease. So we've been using a whole suite of biomaterials, including the polymer that you mentioned, and also nanotherapeutics to deliver chemotherapy immediately at the end of the surgical procedure.